Hello, my name is Sean Go, and my chosen article is Regulation of Fasted Blood Glucose by Resistin. Now, I'm sure many of you know obesity and diabetes are both problems that have been on the rise in the United States. It has been for many years now. For the article's purpose, we'll be focusing on type 2 diabetes mellitus, which is characterized by the impairment or failure of cells to respond to insulin, which helps drive glucose into body cells. The body can resist or ignore insulin for a variety of reasons, but the scientists responsible for this article are very interested in the link between obesity and insulin resistance. Now, I know that if I told you that there is a strong relationship between obesity and diabetes, you would probably say, well, duh, or obviously. But the scientists here are concerned with the interactions between adipose tissue and insulin. These interactions are often the target of many drugs and treatment plans for diabetes. So, adipocytes, or fat cells, in the adipose tissue can act as signaling molecules that will affect glucose homeostasis, usually things like blood glucose concentration, for example. One of these signaling molecules is resistin. When resistin is released, the liver breaks down glycogen to release more glucose into the bloodstream. The researchers have made the hypothesis that resistin's influence on blood glucose concentration also influences insulin resistance on the body's cells. Type 2 diabetes is characterized by high blood glucose levels as a direct result of cellular resistance, or the failure to respond to insulin. Okay, so in order to test this hypothesis, the research group generated mice that are deficient in resistin by replacing the coding exons of the resistin gene with the reporter gene LAGZ. Now, if any of you recall from Bio 181, this is a result of pre-mRNA splicing manipulation, and the replaced gene is LAGZ, which encodes for an enzyme beta-galactosidase. The beta-galactosidase enzyme would be present in the white adipose tissue of the mice, and the absence of circulating resistin could be confirmed using two advanced biology techniques, immunoblot analysis and radioimmunoassay of the rat serum. Two different groups of mice, homozygous dominant and recessive, were placed on a series of diets, a normal diet followed by a very high fat diet, and their blood glucose concentration was measured at regular intervals. Okay, now onto the results. So, for clarification purposes, plus/plus in all graphs means homozygous dominant. Essentially, resistin is expressed in both alleles. Minus/minus in all graphs means homozygous recessive. Resistin is not expressed. The resistin gene is not in either allele. So figure A shows that the body weight gain patterns between both groups of mice were similar. Figures B, C, and D all shows that at just about any given moment that the blood glucose concentration was always lower in mice that lacked resistin rather than mice that had resistin. Figure A depicts a similar weight gain pattern between both groups of mice. Hence, nothing is abnormal with the experimental design. Everything worked just as expected. Figures B, C, and D show that among all times, blood glucose concentration has been consistently lower in resistin-lacking mice than in normal mice, supporting the hypothesis that the presence of resistin consistently increases blood glucose concentrations. So, as you can see in Figure B, when placed on a normal diet between both groups of mice, the normal mice consistently had a higher blood glucose concentration than mice that lacked resistin. Same thing in Figure C with the high-fat diet. Normal mice that had resistin still had a consistently higher average blood glucose concentration than mice that didn't. And in Figure D, following a high-fat diet and a few hours of fasting, blood glucose concentration was consistently higher in normal mice that had resistin compared to mice that didn't. These findings will lead to interesting talks and research about potential gene therapy for the treatment of type 2 diabetes mellitus and the future study of the full function of resistin. These are my references. Thank you for your time.